Thank you for the kind introduction. I'm Eiji Hishinuma from Tohoku University in Japan. Thank you very much for giving me this opportunity to make a presentation today. Today, I would like to talk about the metabolome analysis of plasma using the kit in a large cohort study in Tohoku Medical Megabank project and its application to gynecological cancer biomarker discovery. Although there are various methods to analyze the human phenotypes, such as genome, transcriptome, and proteome, metabolome is most downstream of the central dogma and is considered to best reflect human phenotypes. Therefore, metabolome analysis is considered to be useful in searching for biomarkers for disease prevention and diagnosis. Tohoku University, Tohoku Medical Megabank Organization, TOMO, was founded to establish an advanced medical system to foster the reconstruction from the Great East Japan earthquake. The organization has been developing a biobank that combines medical and genome information during the process of rebuilding the community medical system and supporting health and welfare in the Tohoku area. The Advanced Research Center for Innovations in Next Generation Medicine, INGEM, was established in 2017 as the hub for next generation medicine to bring together the university's preeminent research capabilities in fields such as information science, basic life science, and genomic medicine as the core subject. INGEM has established a clinical biobank where biological samples of patients with various disease, mainly from Tohoku University Hospital. And we are conducting a multi-omics analysis of these patient-derived samples. Previous metabolome analysis in TOMO using mass spectrometry is shown this slide. We used to perform metabolome analysis using global metabolomics and GCMS. But since these reflect relative values, it is very difficult to compare batch to batch and between facilities. Recently, however, biocrates has developed a targeted metabolome kit, absolute IDQ P180 and MXP quant 500. And we have used it to perform metabolome analysis on a number of plasma samples. And the features of the MXP quant 500 kit are shown. And these kits are very useful for our large cohort studies because they can quantify a large number of metabolites in a very high throughput manner. Furthermore, the amount of sample used is very small and normalization between batches is easy. This slide shows the analysis flow in TOMO and INGEM. We are using commercially available plasma as our own global quality control GQC, and we are applying GQC to 4 wells per plate. We have performed many plate-to-plate normalizations using the quantitative values of this GQC sample and have obtained very stable results. In addition, NIST plasma is added to 1 well per plate to check the quality of the data. We have measured approximately 10,000 plasma samples from the Tomo cohort and more than 1,000 plasma samples from the clinical biobank for cancer, diabetes, and cardiovascular disease. Other samples such as serum, urine, bile acid, feces, and exosome have also been measured. The effect of normalization using over ZQC is shown on this slide, using glycine as an example. The upper panel shows before normalization, and the lower panel shows after normalization. We normalized by the median metabolite concentration of each batch of ZQC relative to the median metabolite concentration of ZQC for all batches. And we observed the normalization correct for differences between batches. This data compares the quantitative values of representative metabolites using the kit with those measured by NMR. The graph on the right shows the ratio of the respective quantitative values, and most of the metabolites have well correlated quantitative values, while the 6 metabolites shown in orange have more than twofold differences, indicating differences depending on the quantification method. We have published the quantitative values of metabolites in plasma samples of the Tomo cohort obtained using the 500 kit in the Japanese multi-omics reference panel JMOP, shown here. This database can be accessed via the internet to obtain metabolite information as well as genomic, transcriptomic, metagenomic, and pharmacogenomic information. The database of metabolites looks like this slide. The metabolite common name and ID, number of samples, quantitative values, and links to external database displayed in this way. You can see a histogram like this by clicking on the ID. Some metabolites change in relation to age, gender, and BMI. For example, there is a tendency for CE25 to correlate with age, and for creatinine to correlate with gender. In addition, the quantitative values of NIST and ZQC plasma measured by us are also published in this database. Here is a graph that visualizes the quantitative values of the Tomo cohort samples. The left shows the differences by gender, and the right shows the differences in metabolite profiles for each individual. In the graph on the left, the blue line represents males, and the red line represents females, and the black line represents the median. The graph clearly shows that triglycerides are higher in men, while sphingolipids are higher in women. Profiling and visualizing many metabolites, as shown in this graph, can be used to create personal health care programs and search for predicting biomarkers. In the next section, I will talk about the results of comparing metabolite profiles for Tomo cohort samples and gynecological cancer samples in the clinical biobank. Cancer has been the leading cause of death in Japan in recent years, with 9.6 million patients lost worldwide in 2018. The number of cancer patients is increasing every year, and it's very important disease worldwide. Cancer cells are known to reprogram their intracellular metabolism, and in general, the Warburg effect causes the glycolytic system to become dominant. It has been reported that a variety of characteristic metabolic pathway changes and metabolite fluctuations occur accordingly. Therefore, metabolome analysis is useful for identifying biomarkers for diagnosis and prevention of cancers. First, we present a comparison of cervical cancer and cohort samples. Basic information on the sample is shown in the table. Controls were selected from the Tomo cohort samples matched for age, sex, and BMI. Metabolite with LOD greater than 20% of all samples were excluded from the analysis. And PCA didn't show good separation, but OPLSDA significantly separated the two groups. Cervical cancer samples showed a significant increase in 47 metabolites and a significant decrease in 75 metabolites. A heat map of metabolites that showed significant changes in shown as well. In cervical cancer, there is a marked decrease in lysoPCs. This may have an effect on cervical cancer growth and survival. While decrease in histidine and tryptophan and increase in CE and cysteine metabolites are similar to other type cancers. Also, the AUC was obtained from the ROC curve and was 0.975 even for the top 5 metabolites, which is better than previously reported. Next, the effects of radiation therapy were compared. Significant changes in metabolic index related to unsaturated fatty acids were observed in the non-CR group. This is due to, in part, to the effect of desaturase, whose expression increased in cancer cells. In addition, NOS activity tended to increase in the non-CR group. These results suggest that this method can be applied not only for differentiation from healthy controls, but also for determining radiotherapy sensitivity. The following is an example of analysis of endometrial cancer. As in the case of cervical cancer, similar comparative analysis was performed of endometrial cancer and healthy cohort samples, matched for age, sex, and BMI. The results showed significant increases in 111 metabolites and a significant decrease in 148 metabolites in endometrial cancer samples compared to the healthy controls. The following is an example. The ROC curve for endometrial cancer is shown on this slide. As with cervical cancer, the AUC was 0.997 for the top 5 metabolites with good sensitivity and specificity. The box plot of the 5 metabolites is shown the upper right, showing significant changes between the two groups. Next, we performed a correlation analysis with risk of recurrence and stage. The results showed that cortisol was elevated in relation to both recurrence risk and stage. This may reflect inflammation associated with cancer. On the other hand, the amino acid such as valine, citrulline, tryptophan, and histidine decreased in relation to recurrence risk and stage. These results suggest that recurrence and stage progression of endometrial cancer are regulated by different metabolites. Next, survival curve analysis was performed to determine if these metabolites actually affect recurrence. The results showed significant differences between the 6 metabolites of cortisol, histidine, tryptophan, methionine, alpha-aminoacidic acid, and carnitine and relapse. The results indicate that measuring metabolite in the plasma of patients with endometrial cancer before treatment may predict recurrence. We then compared ovarian cancer samples with those of the cohort. PCA results showed slight separation of the metabolomic profiles of ovarian cancer patients and TOMO cohorts, whereas OPLSDA results showed strong separation, suggesting a characteristic metabolome profile for each group. Next, the results so far suggest that the kynurenine pathway plays an important role in cancer progression. Therefore, we confirmed the relationship between the ratio of kynurenine to tryptophan and the prognosis of these ovarian cancer patients. When the kynurenine tryptophan ratio was compared between the CR/PR and SDPD groups for efficacy of first-line chemotherapy, there was a trend toward higher ratios in the SDPD group, but no significant differences was observed. However, when compared by patient prognosis, the worse the patient prognosis, the significantly higher the ratio of kynurenine and tryptophan. The kynurenine pathway is enhanced in ovarian cancer patients with high IDO activity and may be involved in the deterioration of ovarian cancer by suppressing tumor immunity. Kynurenine tryptophan ratio can be used as a biomarker to predict the prognosis of ovarian cancer patients. Here is a summary of the changes in metabolites of the 3 gynecologic cancers. Metabolites common to each cancer type may be applied as markers for other types of cancers. Monitoring the metabolite characteristic of each cancer may also help to identify the cancer type. So, in conclusion, we demonstrated the usefulness of metabolome analysis using the target metabolomics kit in a large cohort study of TOMO and its application to gynecologic cancer biomarker discovery. In addition, metabolome analysis of patient plasma may be applied not only as a diagnostic marker for cancer, but also for determining treatment efficacy and predicting prognosis. We plan to measure and validate more samples in future. We also expect to measure other methods such as nucleic acid, metabolism, and proteome analysis to identify more reliable biomarkers. Thank you very much for your attention.